This video will cover the topic of enzyme kinetics. Several factors affect enzyme kinetics, including enzyme, temperature, and substrate concentrations, as well as pH, inhibitors, product concentration, activators, and physical agents. The rate of an enzyme-catalyzed reaction is directly proportional to the enzyme concentration. A plot of the rate of catalysis versus enzyme concentration results in a straight line. The reaction rate increases with temperature, generally following a bell-shaped curve. Q10, or the temperature coefficient, is the factor by which the rate of a biological reaction increases for a 10 degrees Celsius rise in temperature. The rise or fall in enzyme activity with temperature is a significant survival feature in cold-blooded animals. In mammals, this assumes physiological importance, seen in fever or hypothermia. Enzyme activity also follows a bell-shaped curve concerning pH, with an optimum pH, usually at a neutral pH between 6 and 8. pH changes affect the charged state of the catalytic site and the conformation of enzyme molecules. Optimum pH varies for different enzymes. For example, trypsin's optimum pH is 7.6, while pepsin's is 2 to 2.5. Acid phosphatase is 5, and alkaline phosphatase is 9 to 10. The accumulation of products decreases the velocity of the enzyme reaction. In biological systems, this is prevented by the quick removal of the product. Activators, such as inorganic metallic cations or anions, combine with the substrate or the enzyme-substrate complex, changing the conformation of the active site. Inhibitors make the active site unavailable to the substrate or change the enzyme structure. Physical agents, such as light and radiation, can influence enzyme activity. For instance, red or blue light increases salivary amylase activity, while ultraviolet light decreases it. Substrate concentration follows a rectangular hyperbola, known as the Michaelis plot. Initial velocity is the velocity when little substrate has reacted. The curve has 3 phases. In the first phase, velocity is proportional to substrate concentration, showing first-order kinetics. In the second phase, it shows mixed-order kinetics. In the third phase, all enzyme molecules are substrate-bound, and velocity remains unchanged, showing zero-order kinetics. The curve's phases correspond to low substrate concentration, 50% saturation, or Km, and high, saturating substrate concentration. The Michaelis-Menten equation relies on the steady-state assumption. The substrate concentration must be much greater than the enzyme concentration at initial velocity rates. In the steady state, the rate of ES formation equals the rate of ES breakdown. This condition is termed a steady-state rate. The Michaelis-Menten equation derivation starts with the steady-state assumption. Rate of ES formation equals the rate of ES breakdown to product. The equation is derived from balancing the rate of ES formation and the rate of ES breakdown, followed by algebraic rearrangement and substitution of the Km and Vmax terms. The Michaelis-Menten equation relates the initial reaction velocity at a given substrate concentration, the Michaelis constant, and the maximum possible initial reaction velocity. The Michaelis constant, Km, is the substrate concentration that produces half the maximal velocity, or Vmax divided by 2. Km is an enzyme kinetic constant, indicating the substrate concentration needed for efficient enzyme function. A low Km means high affinity of the enzyme for the substrate.